Good morning, everyone. Welcome to the MPG Primer. We're excited to have Evan Koch here today. Evan is a research associate in the Sunayev Lab at the Department of Biomedical Informatics at Harvard Medical School. He applies theoretical and empirical population genetics approaches to better understand mutagenesis, variant function, and how natural selection shapes genetic variation in complex trait. He also co-chairs an evolutionary focus group at the NIH's Impact of Genomic Variation on Function, the IGVF consortium. And aside from human genetics research, he also involves himself in research on wolves, Arabidopsis, and various pathogens. He received his PhD in ecology and evolution from the University of Chicago in 2018, and he was advised by John November and Richard Hudson. And today he's going to be talking about introduction to population genetics. And please raise your hand or come up to this mic. I could bring one to you, or you could come here to ask questions throughout or at the end. Thanks.
Thank you. Yeah, so I'm going to give an introduction to population genetics briefly, because it's only a field that's been around now for a bit over a century, so we should be able to cover everything. This is parentheses here, models, so there's a lot of amazing empirical findings, but I'm going to focus on the models because I believe that this provides really the base understanding that we try to get when we look at genetic data from populations. Yeah, so we're going to hit really three topics today, starting with genetic drift as also sort of an introduction into modeling a population generally. Then go on to natural selection, and in particular, negative selection, partly because this is what I actually tend to work on most of the time, and also because it's the one that's most ubiquitous in your genome. And then we'll talk about the coalescent and genealogies as like a sample-focused view of genetic variation and ancestry, and touch a bit on the ancestral recombination graph as like maybe a bit of a hot topic at the moment, and then briefly touch on the extra treat of the selective sweeps in the genome. And then, if there's time at the end, I'll go over a little bit about some tools and resources. Okay, so to start at the absolute, very basic place of genetic drift and genetic variation in populations. So, right, you could have at one position in the genome here two possible alleles, let's say A and G. And what we talk about most of the time in population genetics is the frequency of those alleles, the how common they are in the population as a whole. So if you had a population, let's say, of n diploid individuals, we would just simply calculate the frequency of the G allele by the number of haploid genomes that carry that allele. And so for this talk, I'll denote the frequency of an allele as P, and then the frequency of the alternate or reference allele will always be 1 minus P. Okay, so then let's think about a population. And this is not a very large or realistic population maybe, but let's say you have some number of diploid individuals, and each of those individuals is going to have a genotype being either one of the homozygote or the heterozygote genotypes. And we can then ask what the frequency simply of our G allele is in that generation, right? Then those individuals can pair up and produce some number of offspring, denoted here in the second generation as their children. And given that, we can simply follow Mendelian segregation to, this is an inherently random process, to choose genotypes in the next generation. And so this combination of variation in the number of offspring that parents are going to have, as well as the inherent variation due to Mendelian segregation, are going to mean that the frequency of that G allele is not exactly the same in this next generation as it was in the previous. And this is really what we mean by genetic drift, that there's these inherent factors to Mendelian segregation and breeding structure of populations that leads to variation in allele frequencies over time. Right. So how do we want to, now we want to actually model this? What are we supposed to do? One option would be to try to be realistic and actually specify what the pedigree or some pedigree generating process is in the population. This is an example of a couple of wolf packs in northern Italy, and researchers have gone out and actually mapped out their pedigree, which is great. But as you can maybe see from this, it is enormously complicated, and it would be a ton of work to actually model or measure this for every population we want to look at, and mostly impossible. But we could do that, and then simply follow Mendelian laws to see how allele frequencies change. We're not going to do that. That's way too hard. But what we will do is come up with a simple model of reproduction that we can actually work with. And there's a couple of these that are tractable and have a lot of work done on them, but the most common is probably what's called the Wright-Fisher model. And so that states, okay, we have some population of N diploids at generation P, and then we're going to choose a next generation of individuals at time step T+1. So this is discrete generations. And then each of these individuals in the next generation needs 2 copies of their genome, 2 copies of this allele, and they're going to sample it at random from the individuals in the previous generation. And therefore, we have the variance in the number of A alleles is simply going to be a binomial distribution with simply the probability of getting that A allele equal to the probability that it was in the previous generation. And there's going to be no average change in allele frequencies, no directional change. The expectation of this is going to be 0, but you're going to have this genetic drift due to that sampling process. Now, if we instead calculate the variance in the change in allele frequencies, this is going to be equal to P times 1 minus B over the diploid, twice the diploid population size, right? So we can, just from this very, very simple model, make a sort of qualitative statement about how drift should work. It should be faster in small populations when that N is small, and it should be also faster at high minor allele frequencies. So that variance in allele frequency change will be fastest when the allele frequency is around 50%. But what should we make of real populations? We don't want to do the complicated thing and think about pedigrees all the time. We don't actually believe that a Wright-Fisher model is an appropriate one for, say, the human population. So what's commonly done is to try to make an analogy between the drift that occurs in idealized populations like this Wright-Fisher one and real populations that might exist in the wild, and by matching the rate of genetic drift. And so we introduced this quantity called the effective population size, or NE, and we want to choose the effective population size such that the variance in allele frequency from one generation to the next matches this equation for the binomial sampling in the Wright-Fisher model, where we've substituted in an effective population size instead. Now, one aspect of this is that when we're choosing some alternative model, some real model that we think is maybe more realistic, trying to analogize that to Wright-Fisher ones, the variance needs to actually be proportional to P1 minus P for an effective population size to exist. Thankfully, that's largely the case, but it's something you might consider. Great, so let's maybe take a silly example of this and think about some model where you have a distribution of family sizes, sort of similar to what I showed previously. This model over here with a variance of 2 is going to be approximately equal to what you would see in the Wright-Fisher model, but you might have populations with a lower variance or a much higher skew in the family size that individuals might have. And if you set the number of individuals in the population to be constant and simulate allele frequencies sampling generation after generation, we can see that indeed the drift in these allele frequencies increases with that variance in family size, as we might expect intuitively. And then in mathematical terms, we can say that the effective population size NE should be 4 times the the diploid size over this variance in family size and a contribution to from Mendelian segregation. Interestingly, you could this also, illustrates the fact that even if there was no variance in family size, you would still get genetic drift in diploids just from Mendelian segregation. So then if we went through and we actually scaled the population sizes in each of these family size models here so that they should have the same effective population size, you see that indeed the distribution of allele frequencies after random sampling one generation does actually look very similar. Okay, so one thing that will happen, right, we have this sampling of allele frequencies generation after generation, is that we let that go on for long enough, you're going to eventually remove all the genetic variation from the population, and the alleles are either going to, drop out of the population, go to a frequency of 0, or they might fix in the population and go to a frequency of 1. Now, we know that there is genetic variation in population, so we need some mechanism to add this back in. So that mechanism is obviously mutations, which generate genetic variation that then might be able to drift up to higher frequencies and maintain a balance between the addition of genetic variation into population through mutation and drift, which is going to tend to remove it. So then we're interested in questions of how that genetic variation is maintained and what is its distribution within populations. This provides a nice opportunity to introduce a second approximation. So we can think about this Wright-Fisher model. We want to get even simpler, actually. So we're going to now approximate that allele frequencies aren't this count in the population. We'll say, okay, well, they're actually approximately continuous, which unless you're a very, very rare allele is going to be pretty good a population, which is sort of similar to saying the population size is large. And this diffusion approximation, the dynamics of those allele frequencies are only really going to depend on the expected change in allele frequency per generation and the variance in allele frequency change. And as we've already seen, we already know what the variance is. It's this P1 minus P over the effective population size. So now you can apply this diffusion model and say, starting with some continuous distribution of allele frequencies at whatever starting point you want, go an arbitrary number of generations into the future and get what's going to happen to that distribution. We're going to call that distribution of allele frequencies psi here. And what's going to happen is you're going to lose alleles at the 0 and 1 boundaries, and that distribution is going to flatten out as well. And this evolution happens according to this diffusion equation here, which you don't really need to think too much about right now for the purposes of this talk. But you can see that it depends on this expected and variance in the allele frequency change, as well as what the shape of the genetic variation is at the given point in time. Okay. Yeah, so now if we ask for, okay, if we come to this balance, we come to this equilibrium, what is the distribution of genetic variation going to look like? And so we can add mutation back and forth between our two alleles, set a mutation rate here as the rate per gamete per generation, and then the expected change in allele frequency is going to be mu, this per generation mutation rate has 1 minus 2p. And so, at the boundaries, if that allele isn't actually present in the population, mutation's always going to add at rate mu to the population. And this is the distribution that one gets. It depends only on a compound parameter, which we call theta, which is sort of like a parameter capturing what the neutral genetic diversity should be in the population. And that depends on the product of the effective population size in the mutation rate. And we can also see that most variants are going to be rare, because when you get a variant added to the population by mutation, the most likely thing that's going to occur to it is that it will then just subsequently pretty quickly be lost. But some amount of the time, variants are going to drift up to high frequencies and add this real genetic variation to the population that affects things like complex traits and all other kinds of good stuff. Now complicating things just a little bit further, this is maybe in the previous slide a model for a single site. Genomes are large, and we don't necessarily want to model it site by site all the time. So one trick that we can do is use what's called an infinite sites model. And so now we have this stretch of genome here, and at each site there's going to be some ancestral allele. Mutations are going to create new derived alleles at some positions there, and occasionally they're going to drift up to high frequencies, and you'll have some then pile within this stretch of alleles at some appreciable frequency in the population. And so what you can do is at any point in time take all of those derived alleles that are segregating, having these drift processes at each point in our pile of sequence, and add them all up and ask what sort of the aggregate distribution of those allele frequencies is. And this is what's called maybe the allele frequency spectrum, psi here. And that's going to be the number of sites that we've considered in our stretch here. It's going to be proportional, the number of mutations that you're going to see in each of these frequencies proportional to this theta, this sort of genetic variation parameter, and it's going to be proportional to the inverse of the allele frequency. Right, and so this, we're going to assume most mutations are rare events. We can model this as a Poisson process, and at the diffusion, the different positions are just going independently. And so now we can make really nice calculations from this allele frequency spectrum object, such as just calculating what's the expected number of derived alleles we would expect to see in some frequency range. And that's going to be Poisson distributed with simply this, how much mass is there in this frequency range according to this allele frequency spectrum object. Great. So now we have this model for these latent allele frequencies in the population as a whole, but a lot of the time what we're working with is a sample from the population. We've sampled some genetic sequences, right? And what we're going to do is calculate what's called the sample frequency spectrum, or the SFS. And so what we're going to do then is for each possible count of alleles in the sample, see how many observations we make at that count, right? So if we sampled 6 sequences here, we would count up the number of alleles that we saw at a frequency of 2. So you have 2 of those here, the number at frequency 1, and so on and so forth, and add that all up and ask how many times we saw alleles at each possible derived count. Now we can develop a statistical model of this SFS using this allele frequency spectrum. Let's say if we took a sample of n haploid individuals from the population, simply by applying a binomial sampling from that latent distribution of allele frequencies. And this has the nice property of being Poisson as well. And so you can make very simple calculations just from having this allele frequency spectrum object, applying this binomial sampling to it, and then getting a model for what the distribution of allele frequencies should be that you expect to find in your sample from some population with a particular frequency distribution. And that looks something like this here, this black line being the latent frequency spectrum, and then these blue dots being the expected counts that you would get in a sample. And so, if we sampled some variant and found it at, let's say, frequency 0.5 in the population, we can ask simple things like, well, what's the latent distribution of allele frequencies that 50% allele actually represents? And there's actually, for a sample of size 10, there's going to be quite some variance in that, and this is going to shrink as as your sample size gets larger. But you might also note that for low frequency alleles, a lot of the mass of what the latent allele frequency is going to be substantially lower than what you actually saw in your sample. And so you're going to find those rare alleles that you kind of catch by accident in your sample at a little bit higher frequency. In real populations, in human populations, let's say, the SFS doesn't look like this 1 over P curve. So here, this is from Nomad. For non-Finnish European individuals, this SFS object for synonymous sites. And here these dashed lines show what one would expect from this stable, constant size population that we've just been talking about. And we can see that the SFS is a lot steeper. You have an excess of rare variants, and it's very far from this standard neutral SFS of 1/I. And this, one way that this happens, and you probably have other factors that affect this as well, is that the human population size hasn't stayed constant over the past, whatever huge amount of time. And so one thing that people do is fit demographic models using this site frequency spectrum. And so this is just one from Tennyson et al. For European populations, largely kind of trying to approximate some sort of out of Africa events. So you have assume that there's some stable population size at some long time in the past, denoted by this dashed line here, and then evolving that through a population size trajectory generation after generation in this diffusion model, and then getting what the allele frequency spectrum is at the end of that whole thing. And that would be this black line here for this Tennyson model. And so you get like various features in here, such as this dip in genetic diversity due to out of Africa bottleneck, as well as a large number of rare variants due to population growth. And one thing I just want to sort of impress you that all of this is I hope it doesn't sound too complicated, and please again interrupt me if you have any questions. But it's actually quite simple to start doing yourself. Nicely with this program, Daddy, you can simply in Python set up a frequency grid, get your equilibrium spectrum, and evolve that spectrum with like one line of code for some number of generations, and then ask what a sample would look like. And it's very easy to start playing around with. And then you can start making calculations for what you might expect in your sample, what might you expect from selection and heritability contributions. And it's very fast to do. You don't need to do complicated simulations. You can get these allele frequency spectrums super easily. Okay, so now let's get on to natural selection. Okay, so when we talk about selection, what we're talking about—
Hey, Evan,
Yeah.
Before you move on to your next step, we had a question online from Sudeep. He says, we know about conserved genomic locations. Is this knowledge accounted for in the model?
I'm assuming you mean like demographic models. And yes, to the extent that when people try to fit these demographic models, they try to find the most neutral regions of the genome that they can. So they're not going to use like missense mutations. They'll use intergenic or synonymous sites to try to be as unaffected by selection as possible. Yeah. 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 Great. So what we talk about when we talk about fitness or natural selection is allele frequency changes that are due to differences in fitness among genotypes. And we can define fitness pretty broadly as the reproductive contribution of an individual to the next generation. And then we would assign fitness values to each of the possible genotypes at our site. And we can kind of think of these as average values for each genotype. Then the allele frequency change due to selection in each generation, the expected allele frequency change, is going to be proportional to P1 minus P, so this sort of genetic variance at that locus, and the regression of this fitness value on the genotype, right? So we can code these genotypes as like 0, 1/2, or 1 by the frequency of the allele within that genotype. And then the regression of fitness on that is going to tell us what should happen in that generation on average to the allele frequency. And so things can be additive if there's simply a linear relationship between genotype and fitness, or you can have more complicated dominance relationships, where those fitness values don't necessarily have a more complicated relationship, let's say, with fitness. Yeah, so right, a lot of the genome is constrained, as the question asker had asked. So there are various estimates of this, but let's say that like maybe approximately 7 to 10% of the human genome is constrained by what we would call negative selection. This can vary a lot from coding regions where it's really, really high to transposable elements where it's almost entirely absent. But a large proportion actually of the human genome is subject to some amount of negative selection. And we're going to very simply model this by saying, like, okay, we have some ancestral genotype, let's call it this A. And then the heterozygote and homozygote genotypes are going to have some reduction in fitness due to selection. And for simplicity, just let these be S and 2S to do this additive relationship. And if that's true, the expected allele frequency change should be proportional to that selection coefficient S here times this P1 minus P, right? So now we can again apply this diffusion model and a Poisson random field, infinite sites approximation, simply as we did before. And just like we got this theta, this any times mu parameter out the first time, you add this second parameter, which I like to call big S, which is the product of the effective population size and that selection coefficient, right? And so this gives us then something we could call, if purifying selection is not too strong, some mutation selection drift balance, where as this compound S parameter increases, you go from the neutral site frequency spectrum, right, which is this linear relationship with derived allele frequency and the frequency spectrum, and you start dropping off as you reach higher frequencies. And so we can conclude that as selection coefficients go up, that natural selection prevents you from reaching higher and higher, or as selection coefficient goes up, natural selection prevents you from reaching increasingly low allele frequencies. And one way in which this is conceptualized is as a drift barrier that at approximately this 1/S frequency, selection will prevent that deleterious allele from getting any higher. And so that's drawn as these dashed lines here. And this actually provides, in many cases, a pretty good approximation to the action of selection in the case of weak purifying selection. Right. So one observation that's come out of a lot of work in human genetics and in other organisms as well is that actually a large amount of human mutations generate this weakly deleterious variation, right? We have all this constrained sequence in the genome, but we know that it's not anywhere close to lethal because that would be actually quite bad. You're not getting there. Right, so we can use the SFS actually to estimate the distribution of this weakly deleterious mutation. We have this sample frequency spectrum SFS over here. We previously, integrated over some binomial sampling, right? But we can additionally add to this distribution of fitness effects or DFE that's hopefully going to tell us how much of the genome is creating selection at different intensities. And this is done by comparing these minor allele frequency counts for different regions of the genome. So intergenic sites have more genetic variation compared to even synonymous and certainly non-synonymous sites. And you can use that to estimate this distribution of fitness effects. When this is done, you do indeed find this huge mass of mutation that should be generating these weakly deleterious mutations. These weakly deleterious mutations are of pretty high importance because they contribute disproportionately to trait and disease heritability, as well as the GWAS hits that we actually find. So if we say that like the contribution to additive genetic variance for an allele at frequency P is this 2 beta P1 minus P. And if we let the selection coefficient, let's say, be proportional to, let's say, the squared effect size, then you can calculate what the expected contribution to genetic variance should be from mutations with selection coefficient S. So if we plot the per site contribution to that genetic variance against the selection coefficient, we can say that it increases with the strength of selection. And that's even higher if you assume a model of stabilizing selection, versus purifying selection, which I won't actually really explain, but let's suffice to say that like a large amount of heritability should be contributed by deleterious alleles, and it might even be more in this nearly neutral range than expected from a simple model. And indeed, when this is from Yuval Simons and Jonathan Pritchard's group, trying to estimate that DFE for GWAS hits for a large number of different traits. And again, indeed, you do find this, large mass of heritability that should be coming from this intermediate range of weakly selected variants. This also makes sense in terms of a phylogenetic constraint, where if we look at phylogenetically, trying to find phylogenetically constrained positions in the genome, that is, places that don't vary very much between related species, such as mammals or primates, and annotating variants by this constraint in phylogenies shows that this is, perhaps by far the largest contributor to heritability enrichment of any kind of annotation you might provide, right? And so, how does that then coincide with our view of weak selection contributing a lot of heritability? Well, if we ask how often should mutations fix relative to neutral for different strengths of selection, we can see that you don't actually need that strong of a selection coefficient in order to really prevent almost any fixations to occur on this phylogeny. So if we're talking about getting to constraint up in this area on the right over here, you very quickly get there. You very quickly get to this nearly neutral range. Okay, so that was weak selection. On the other hand, selection might actually be very strong, and you can sort of almost remove the drift part of it to get to what we might call just mutation selection balance, where at equilibrium, the expected frequency of strongly selected mutations should be the mutation rate over the selection coefficient. And right, this sort of makes intuitive sense that as mutations actually become lethal, so as heterozygous selection coefficient goes to 1, their frequency just match the de novo mutation rate, right? And in reality, there's a variance in this that's determined by this theta parameter. But for many applications, thinking about things in terms of this deterministic mutation selection balance is actually really helpful. Right, so now we want to measure strong constraint in the genome. What are we going to do? Well, a large fraction of the genome has mutation rates that are on the order of, let's say, 2 × 10⁻⁹ per generation. And so if that's close to lethal, that's like your frequency in the population. And that would require an enormous sample size to say with any confidence that this mutation was actually having these strong fitness effects. And so we still need to group sites in order to estimate the proportion that are under this extreme purifying selection. Where this is most commonly applied, perhaps, is in the case of selection on loss of function mutations in protein coding genes, where in addition to protein truncating variants, you have a bunch of stuff that affects splicing. And then you can then apply a model where, let's say, we've estimated the mutation rate at each site in the genome, and you can get this mu parameter in order to estimate that selection coefficient. Yeah, this is what's done sort of heuristically in this Loof score, maybe just by comparing observed versus expected counts of loss of function mutations, taking that lower bound there. And indeed, like when you calculate these constraint measures, they're highly enriched in haploinsufficient genes and not in neutral stuff. Alternatively, there's methods that actually explicitly estimate this heterozygous selection coefficient. And interesting, you might ask, okay, well, am I really getting at this? Am I really getting the calibrated S hat, or am I just getting something proportional to it? And it does actually seem to be the case that you're getting something that's on the correct scale, because one prediction is actually that these S hat estimates, if they're real, should be approximately equal to the fraction of mutations in any generation that are caused by de novo, the fraction of variants that are caused by de novo mutations in any generation. And this does seem to line up pretty well, indicating that these S hat estimates are actually somewhat estimating that real population genetic parameter. Yeah, and there's some available estimates of this. I won't go through that right now, though. Okay, so now I guess in the remaining time, we'll get through as much of the coalescent as we can. And this is really going to change gears a little bit, but it's another way of looking at genetic variation that's extremely valuable. Right, so we have, a simple Wright-Fisher-like population here on the right. And in each generation, individuals are going to, contribute some number offspring to that population. You can go through that generation after generation, and this is what we thought about as creating this genetic drift, right? But alternatively, we can view this as, let's say we sampled some individuals into the present and we followed them backwards in time instead. And so each generation, we're then going to follow that variant to the parents and doing that step by step. And eventually what's going to happen is that pairs that we sampled in the present are going to find their genetic variants in the same parent at some point in the past, and meaning that they, inherited from that parent the same genetic material. And this is what we call coalescent events. And if we're looking at things backwards in time, this is where those independent lineages actually merge. And eventually you're going to get back to just one single lineage as sort of some like grand ancestor of the population, at the very least at that locus. And so there's two things about this. We want to model backwards in time, but we also want to think about genetic variation in terms of genealogies, which is actually something that's true regardless of what model of evolution one assumes. The most common statistical model for this is called the Kingman coalescent, and it's a simple model for building the genealogies. Totally agnostic to any mutations, we think about the genealogy first. And so going back to this example, we had these three samples here, and we had their coalescent events here for individuals 1 and 2, and here for individuals 1, 2, and 3. And all that really matters is this very simple tree here on the left. There's a ton of evolution that happened in the rest of the population, but if we've just sampled these 3 individuals, that doesn't have any effect on the patterns of genetic variation in our sample. So we can really just focus on what happens to the lineages corresponding to these 3 individuals. So the generating process for these under what's called the Kingman coalescent is to say, okay, we've sampled some, let's say 4 lineages from the population. Each pair is going to coalesce at a rate inversely proportional to the effective population size. And that's going to be, so then we would sample a time to a coalescent event. These are going to be like competing exponential distributions. And then when that coalescent event occurs, we just choose a pair of lineages to coalesce. And you can just apply these rules again and again to generate random genealogies. Then the nice part of all this is that the genealogical process is totally independent of the mutational process. So you can sample your genealogies, and let's say we sampled this tree over here for these 4 samples, and then you can add mutations on top of that after the fact. And you can really choose whatever mutational model you want. We might use the infinite sites model as we had previously and say that mutations occur as a Poisson process along each branch with some rate mu. And under this model, each mutational event would correspond to a unique SNV, which is again the exact same model that we considered previously with this Poisson random field and the site frequency spectrum. But you could also put in some whatever nucleotide substitution model you want. And the important part is that these neutral mutations are independent of the genealogy. We can add recombination to this. Recombination is a very complex process, and we're going to model it as some probability of recombination that occurs each generation with some rate R. And so the total recombination probability for some sequence L would just be L times R. Now, in addition to these coalescent rates, if we're viewing things backward in time, we can also add in recombination events that are going to split pieces of your sampled genetic material up into independent lineages. So you then have this process where lineages are both splitting up due to recombination and coalescing due to shared ancestor events. And so this is a very simple example here. First, you get this recombination event that splits this up. Then this piece of the genetic material coalesces with this other sample such that you've found a common ancestor event for that first piece of the sequence there. And then eventually it's going to coalesce with the rest of the sequence. And what this would lead to is a shorter gene tree at this first part of the sequence and a longer one at the second part. This leads to correlated genealogies along the genome. right? So you're doing these recombination events. It means that the genealogies at nearby positions, even with recombination, are going to be correlated. And this is how we get, correlation of genetic variation and linkage disequilibrium. So one way to then summarize all of this, complicated process, the set of genealogical trees at each locus, is through the ancestral recombination graph, which really says, right, this is the sequence of genealogical trees at each locus in the genome, these recombination events that connect them, and the locations and identities of mutations. And this is an object that's, really a focus of a lot of recent research, and there's a large number of models that are trying to estimate this thing for your sample at each point in the genome. There's two common features of ARG inference. This is a difficult process. One trick is to sort of cluster your samples at each point in the genome to then, get a position-specific distance matrix between all your haplotypes. And then use some clustering out and produce trees. And another trick is to sort of, if you have this ARG, you can thread new haplotypes onto those existing trees. And sort of combining both of those two things, you can get actually pretty good estimates of the ARG genome-wide. So quickly for selective sweeps, just to touch on positive selection. Okay, so why is this in the coalescent part? Well, one of the main features of sweeps is that they have this effect of selection on nearby genetic variation. So if we had a positively selected allele, it's going to have this sort of like logistic growth trajectory up from low frequencies to high ones. And the rest of the neutral variation linked to that selected site is going to have to sort of follow this size trajectory, right? And so that's going to cause longer branches, leading to more low frequency variants, but also reduce genetic variation. They're going to have to coalesce. Lineages are going to have to coalesce before the origin of this mutation. You compare this to just like a neutral genealogy and make some inferences about where selective sweeps occurred in the genome. Right. So again, okay, so you can have some example. This is just from a silly simulation of what genealogies look like linked to a sweep versus neutral ones. You can see there's a lot of this really rare variation down here, and you don't have as much of these long internal branches with lots of descendants, right? And so methods try to estimate sweeps in the human genome and elsewhere. And it turns out obvious sweeps are quite rare in humans, so there are certainly a large number of high confidence samples. This is from this Swiffer method from the Ramachandran group. But in general, like, sweeps don't seem to be particularly common. And if you look around synonymous versus non-synonymous substitutions, the patterns of genetic variation are actually quite similar, indicating that there hasn't been these massive flux of sweeps across human history. Okay, so I guess right before we end, I'm going to talk a little bit about simulation. So we've gone through a lot of models, and one thing I want to impress on you is just try to make calculations under these yourself. And when you do so, start as simple as humanly possible. And so if what you want to calculate is just a summary, some simple summary of allele frequency, using simulations like these DADI diffusion models is really great. You can incorporate negative selection. It's incredibly fast. You can incorporate complex demographic models and get this allele frequency spectrum that you can then make your downstream calculations from. If what you want is neutral mutations and you really care about modeling linkage disequilibrium, coalescence simulations are the way to go. You can quickly simulate very large chromosomes, for instance, with this MS prime method. You can specify recombination rate and mutation rate, complex metapopulations, and even these effects of selective sweeps on neutral variation. And if you really, really need to go more complex than that, you have interaction between multiple selected sites and their effect on neutral genetic variation, as well as their own frequencies. Then you might have to do Wright-Fisher simulations, such as in this SLIM framework. This is necessarily slow because it's simulating all of these processes generation by generation. And you can simulate stuff like polygenic selection, multiple variants, but it's very easy to make mistakes and not realize that you did so. And the manual is 850 pages long. So I would encourage you to start simple if you can. Yeah, I guess we're about out of time. I have some slides on some resources and genetic data, but those will be posted online, I suppose.